Students, in previous lecture we get detailed information about mitosis cell division. Then today we are going to learn about the second step of cell division, meiosis. This meiosis occurs only in the germ cells. Means meiosis is necessary for gamete production and also spore formation. Gamete production means there is a formation of male gamete and female gamete. And spore formation means there is a formation of spores in case of plant. Here we see the one plant moss and there is a formation of spore. From this we can say that meiosis plays a vital role in reproduction process. In meiosis, from one diploid cell, there is a formation of 4 haploid cells. Diploid cells means it have 2n number of chromosomes and haploid cell means there is a n number of chromosomes. Meiosis is completed into 2 stages, Meiosis First and Meiosis Second. Let's start Meiosis First. This process completed through several steps. Look at this picture. There are different stages of meiosis first, in which prophase first, metaphase first, anaphase first, telophase first. And also we see here the prophase first is completed through these 5 processes or 5 stages. That is leptotin, zygotin, pachetin, 4th diplotin and 5th dikynetin. Then see in this picture, during meiosis first, crossing over occurs in the homologous chromosomes. These homologous chromosomes are present in the mother cell. This mother cell is in diploid. Mother cell is a diploid. Diploid means it have 2n number of chromosomes. Then what is meant by homologous chromosomes? We learn this into the 9th standard. The homologous chromosomes are the chromosomal pairs that are similar in length, gene pair and centromere location. Look at the picture. There are homologous chromosomes which are same in shape, size, type and same gene pair. Also the location of centromere is same. See the second figure which shows there is a crossing over between homologous chromosomes and thereafter there is a genetic recombination between these homologous chromosomes. The crossing over result into genetic recombination and thereafter these chromosomes are divided into 2 groups and finally 2 haploid daughter cells are formed and each one has n number of chromosomes, which means each cell possesses of only one copy of chromosomal pair. In this way, meiosis first is completed and then after there is a second stage in meiosis, that is meiosis second. This meiosis second is just like a mitosis. We learn the mitosis cell division. In meiosis first, there is a formation of 2 haploid daughter cells. In meiosis second, the 2 haploid daughter cells which are formed in meiosis first undergoes a division and finally 4 haploid daughter cells are formed. Means from 2 haploid cells there is a formation of 4 daughter cells. These 4 daughter cells contains n number of chromosomes and the mother cell contains 2n number of chromosomes. Since there was genetic recombination in meiosis first, all the 4 daughter cells are formed are genetically different from their mother cell and from each other. Means these daughter cells are different from their parent cell. Also they are different from each other. These cells are transformed into gametes which are always haploid, that is have n number of chromosomes. Then let's see the significance of meiosis. We know the meiosis is very important in reproduction process. In reproduction, during fertilization, the male haploid gamete means sperms which are unite with female haploid gamete, that is eggs, and there is a formation of diploid zygote, means two haploid cells combine and there is a formation of diploid zygote which divides, means this zygote divides and grows into new diploid living organisms. Therefore, formation of haploid gametes help the living organisms to maintain the number of chromosomes in its species. This is the significance or importance of meiosis. After learning about these cell divisions, let us do an interesting activity, that to observe the different steps of mitosis in an onion plants. Let's start the activity. Take a medium size of onion and keep it into the glass jar filled with water in such a way that the roots of onion remains dipped in the water. Then the new roots develop within 4 to 5 days, and after that cut the tip of these roots and put them into the petri dish. Add few drops of iodine solution. Then take one tip of the root on the glass slide and press it with the help of forceps. After that, add 1 to 2 drops of water on it and cover with the cover slip. When there is a completion of preparation of slide, then observe this slide under compound microscope and see which steps of mitosis we see in this slide. Then we see the metaphase step in the cell of the root tip. How did you to identify this step? As we see, the chromosomes are arranged parallel to the equatorial plane of the cell. We can say the step observed is the metaphase step. After that, you can sketch the figure in your notebook. In this way, we learn the cell division is completed through the two stages, mitosis and meiosis. Also, we completed the second chapter, life processes in living organism, part first. Thanks for watching all the videos. If you like the videos, please subscribe our channel and press the like button and share the link to others.